So without further ado then, it is my pleasure to start this webinar and to welcome Professor Thomas Bjarnsholt and Dr. Mads Lichtenberg to SimCell to talk about their research. This is the second webinar in the series, and it's going to be fantastic to hear from these two exceptional scientists in the biofilm field. So they come from the Costerton Biofilm Center at the University of Copenhagen, and I think it's fair to say that the research and the work that is done here is synonymous with really pushing the frontiers in biofilm research, and also making us think and question and relook at what we talk about when we talk about biofilm, especially in clinical settings and chronic infection. So today I'm really excited to hear about their new research titled Metabolic Fingerprint of Pseudomonas aeruginosa as single cells, but also in biofilm. And with that, I would like to stop sharing and hand over to Professor Thomas Bjarnsholt. Thank you, Thomas.
Thank you so much, Ben, and thank you, SimCell, for allowing us to present our data here today. And before we get to the interesting part, which Mads will present, I'm just gonna tell you a little bit about, in our perspective, what a biofilm is in vitro and in vivo, and also why it's important to maybe distinguish between these two habitats. But also, first of all, when you hear talks about biofilms, also when you read about it, we often talk about biofilms as the most common way of bacteria being present everywhere in nature, but also in human beings. You have an example of stromatolites in Australia, 100 years of old, big mats of bacteria assembled, but also here in a creek where you have this slimy surface on top of the rocks embedded in the creeks. And that's actually why it's called biofilms. It was Costerton who was examining the surfaces of the rocks and creeks in the alpine creeks in Canada, and he noticed this biomaterial, the bacteria on the rocks as this slimy surface, and thereby a biomaterial on the surface. That's why it was termed biofilms. But also it's in the way of us human beings. Bacteria can form these aggregates and surfaces, and also in different tissues. Here's an old water pipe. So in 50, 100 years, bacteria can mineralize the minerals in the drinking water and form these huge mats in fresh water pipelines. Whenever we insert something in the human body, any processes or anything, bacteria can form biofilms on it and also adjacent to the implants. The plaque on your teeth is bacteria, can cause caries and so on. Any vessel submerged in water, bacteria and algae form on it, forming biofilms. In the patient suffering from cystic fibrosis, the chronic infections here, the bacteria are also situated in the mucus as aggregated bacteria, and also within the chronic wounds. But one thing to be very good take-home message of this is that these are not biofilms you see here. These are habitats of biofilms. You can see biofilms with your naked eye. And also the question is, do we understand biofilms at all? We know it's some kind of physical aggregation of bacteria. We know matrix of binding the bacteria together. We know this from in vitro observations. We know the matrix can consist of extracellular DNA, proteins, and polysaccharides. But in the human body, in infections and so on, we do not actually know what the bacteria produce. We do not know if the bacteria are producing matrix components at all. That is actually also something we are investigating in the lab. But we do know that there's a striking difference between aggregated bacteria, biofilms, and also the bacteria we can grow in shaken cultures in the lab. This movie was actually supposed to move, but these are the planktonic bacteria, the fast-growing bacteria in exponential mode. But to understand biofilms and also to investigate biofilms for the last 30 and 40 years, we have had a tendency also in our lab to simplify and reduce. So we know that within chronic infections, such as this chronic foot ulcer, we have the bacteria situated in aggregates, as you see here in this picture, where we have stained the bacteria with the FISH technique present in situ hybridization using PNA probes. So we know the bacteria aggregate in the wounds. Because the bacteria aggregate in the wounds, we say, okay, so then that could be similar to the biofilms we grow in flow cells, these mushroom-shaped structures. And then we correlate that then bacteria in wounds, or the bacteria on surfaces, are the same as the bacteria in wounds. So we draw the simplification and conclusion that a biofilm in a chronic wound, first of all, starts with planktonic bacteria, and also maybe you can actually find these mushroom structures in the chronic wounds. But that probably is not the case. And the consequence of this is that there's been a tendency that we can grow bacteria in flow cell systems, but also in microtiter plates. And due to that, we say, okay, so if we can grow the bacteria here, the biofilms are the same in the chronic infections as in the flow cells in this model system. Then whenever we want to test something, we can just do it in the microtiter plates. And that is problematic because the microenvironment is completely different in these two scenarios. In flow cells and in microtiter plates, we have bacteria attaching to the surface and building these mats of biofilms. And then you will have gradients going from the outside and in. So we have fast-growing bacteria outside because there's more oxygen and nutrients for the outside bacteria. And then we have subpopulations because it's only the different layers of bacteria causing the gradient. So we have an inner subpopulation not growing but susceptible to colistin, and an outer layer growing susceptible to topramycin and ciprofloxacin and so on. But the microenvironment in the chronic wound is different because here we have the surface which cause already from up here the gradients of oxygen and nutrients, and then the fast growth is only on the surface. Then we have slow growth or no growth at all within the wound. So we have a lot of dormant bacteria, a lot of tolerant cells, and mainly because it's actually the neutrophils that can actually suffocate the bacteria using the oxygen. But another thing which separates in vitro and in vivo bacteria, not just when they're in biofilms, but also just when they're growing, is that again the microenvironment. And the microenvironment also causes the bacteria to have different expression profiles. And that we elucidated together with Marvin Whiteley. We published this paper in PNAS, and here we showed that there's a huge difference between the gene expression between in vitro samples compared to these 15 human samples ranging from the chronic infected cystic fibrosis patients, chronic wounds, and also acute burns. See from this PSA plot that the in vitro grown bacteria up here in blue are different from all the Pseudomonas aeruginosa present directly in the chronic infections. So again, we have differences between in vitro and vivo, and here it's also the probability it's caused by the microenvironment is at least what we believe. Then also, how does infections initiate? How do we initiate our model systems? So we learned from this mental model that we should initiate our model systems with planktonic bacteria, and also a lot of protocols for initiating animal experiments always with we have to start with planktonic bacteria. But that doesn't really correspond to that we know that most of the bacterial biomass in the environment, also as human beings, are by small aggregates. That we can see, for example, by visualizing bacteria on the human skin. This is fresh and healthy skin, and we can see in these small crevices the aggregates of bacteria, or the bacteria present in the aggregates, and not really as planktonic bacteria. And we can look at the aggregate size of the bacteria on moist, healthy skin, and it's around 10 micrometers. So they're not really present as single cells. But that could also be one of the differences between if we can get chronic infections or not, because we made some, and these are just in vitro experiments, that if we isolate human neutrophils phagocytes, they're actually able to phagocytose the normal aggregate size, these around 10 micrometers. Of course, we can see here from this, you see this from this, these experiments, that a normal phagocyte can phagocytose around aggregates 7 to 10 micrometers. And if the aggregates are bigger, more neutrophils are needed to do that. On the other hand, if we have a skin disorder, then, for example, here this periodontitis suppurativa, a skin disorder. Here we have the bacterial aggregates. They are much bigger, an average of 100 micrometers in diameter, and these cannot be removed by the neutrophils. That could be an explanation if we have too large aggregates, either caused by some dysefficiency in the human skin, or a slow recruitment of neutrophils. Then we can, for example, implant-related infections can initiate, and also chronic infections in wounds and so on. So a summary of this is that we have the normal size of bacteria on the human skin, these 1 to 10 micrometers, are easily phagocytosable. But if something is wrong, so we get larger aggregates, suddenly they cannot just be eradicated anymore. Then also, when we talk about acute and chronic infections, there's always been this saying that acute infections are caused by planktonic bacteria, and chronic infections have been caused by biofilms. But actually, when we look at the classic biofilm infection, like the chronic infection of cystic fibrosis patients, actually much of the biomass are situated either as single cells or very small aggregates. But of course, we also have the larger aggregates, as we see in this microscopic picture. We also have a lot of single cells. So we can't divide acute and chronic infections by planktonic and biofilm bacteria, because they might have, or they both play a role, and that could possibly have something to do maybe with metabolism, as Mads will talk about when he presents his work. So where are the bacteria? Are they, if we insert an implant, are they directly on the surface, or are they in the intermediate zone, or are they in the inflamed tissue? So Louise Kruse Jensen, she has this animal model, a pig model of implant-related osteomyelitis. And they make a drill hole into the tibia of the pigs and insert a stainless steel implant in this implant cavity. And then when we look, or when she looked afterwards, she could find bacteria still stuck on the implant, but also out here in the inflamed bone tissue, or inflamed bone. So the bacteria not only stick into surfaces, but it can also be present adjacent to a foreign object. And also, what is important to notice that, for example, how does antibiotics kill bacteria and biofilms? It's always been that biofilms are much more tolerant than planktonic bacteria, and that is of course also true. But as you see from Louise's experiments in animals, actually just the microenvironment of the biofilm also plays a big role. They took some pigs and again made a drill hole, inserted a implant into the implant cavity, and then had bacteria growing, added bacteria, I had them to grow for 5 days, and then they treat it either with vancomycin or ciprofloxacin for 8 hours. And by microdialysis, they measured the amount of antibiotics. First of all, they could measure in serum, the serum concentrations, but also with these microdialysis catheters closer and closer to where the bacteria are situated, to the implant cavity. As you can see from these two graphs, that the serum concentrations were quite normal. It could reach quite high serum concentrations treating the animals with these two antibiotics intravenously. But close to the implant, hardly any antibiotics could get in. So not only are the bacteria tolerant, but the microenvironment of the infection also causes very, very little antibiotics to penetrate into it. But then, of course, maybe we can just treat, if we insert an implant to prevent bacteria from forming biofilms on the implant or adjacent to the implant, just treat with extreme high concentrations of antibiotics. So Louise, she also tried to add bacteria together with increasing concentrations of gentamicin up to 16,000 times MIC. So they added the bacteria together with the antibiotics, and in vitro, 16,000 times MIC would kill these planktonic Staph aureus very, very easily. But actually, in her implant model, even 16,000 MIC were only able to eradicate the planktonic bacteria in 3 out of 4 of the pigs. So there was one of the pigs where you could still find bacteria left after some days. So again, the microenvironment of the infections really plays a huge role, and also why you cannot necessarily just model bacterial biofilms in vitro without taking these considerations into play. Oh, yeah. So in summary, we have 2 great biofilms. We have the model system. We also have the chronic infections, and both are biofilms. Both are just as good biofilms. There's no biofilm better than the other one, but we need to understand both of them before we can start to investigate the infections in vitro and start to extrapolate our in vitro results into an in vivo setting. And we have a great mental model. It's great for teaching and to understand what is really going on, but it's just a mental model, and it only for certain in vitro flow cell biofilms of Pseudomonas aeruginosa. Actually, Karen Sau, who did this mental model in the first place, told me she never really had the idea that this mushroom structure model should be the role model for all biofilms. And last year we met in Seattle at a biofilm bash where we discussed this, and we decided that we need some kind of better universal model where we have aggregates with and without matrix and host material, but also have single cells that represents how bacteria can be both in vitro and vivo. In vitro, the bacteria can definitely form biofilms, but the biofilm can start both from single cells, but also from small aggregates. And the same also goes for within the human body, both in the healthy and also in the disease situation. We can have bacteria, single cells, and biofilms on surfaces. We can have them within host material, and also have them free floating in liquid in infections. Oh, I have to go right. So in concluding remarks is that we have to start to know, not just to believe. We have to dig into the literature. If you do in vitro experiments, start to look into literature. What is actually known about the infection you're trying to study, and you can't just perform every single experiment in a microtiter plate. Maybe we also need to start to discuss not whether, for example, matrix is present, but the problem of persistence. Why the bacteria persisting? It's probably not just because of the matrix. And whether it's a biofilm, an aggregate, planktonic, single cell might not be so important as to why and how bacteria infect and persist. So we have maybe to take a step back and start to approach it maybe a little bit differently. So this is just it's a very important people, my group here in Copenhagen, collaborators and funding. And also we run these 2 Biofilm courses, both the PhD course, you can look at this Biofilm course, ku.dk, 10-week course, but also we have this free Coursera course on the Coursera platform. We also learn about bacteria and bacterial biofilms. And by that, I don't know if we should take a few questions, or else I'd like to hand over to Mads, where the real fun starts.
Thank you very much, Thomas. I don't think we have so many questions coming in yet, so maybe we can go to Mads straight away and we take all of the questions at the end, if that's okay with you guys. Great, thank you very much. Over to you, Mads.
Yes, hello, can you hear me?
Yes.
Yeah, good. I would also like to thank SimCell for hosting this webinar to allow us to show some of our recent work. And I'll just jump right into it. So I would like to present what we've done the last few months. So we bought the Cal Screener and we asked sort of a simple question, one might say, whether if biofilms and single cells displayed different metabolic different metabolisms. So we investigated this, and I'll just give a little bit of background. So as Thomas also said, bacteria are living either as single cells or in biofilm. And in nature, as he also said, they can form biofilms as large patches covering surfaces and encased in self-produced matrix components. But in infections, you often see these like small non-attached aggregates that are embedded in either host material or intraluminally or so on. So we have a distinction between the large patches and these small aggregates. Okay, and the switch from single cell to biofilm lifestyle is often associated with a change in phenotype and behavioral changes. And a lot of different biomarkers have been developed to describe this switch, such as the increased violence, altered gene expression, physical aggregation, slow metabolism, and increased tolerance both towards host defense and antibiotics. However, many of these factors are not necessarily universal and may change under different conditions. And the only consistent biomarkers of biofilms remain the physical aggregation and increased tolerance. And the routine identification of the biofilm phenotype is often invasive and is work intensive, for example, in the clinic. But it can be assessed, of course, by testing tolerance towards, for example, antibiotics, or by visualizing the aggregates of the biofilm using microscopy. The metabolism, however, has been shown to play a central role in the increased tolerance. So we asked, can metabolism be used as a biomarker for identification of biofilms? And we approached this by growing Pseudomonas aeruginosa, either a single cell culture as a surface grown biofilm or embedded in an alginate matrix. And we pre-incubated these models for either 0, 24, and 48 hours and made these microcalorimetric measurements, which measures the sum of energy related processes as heat flow. And for those of you who don't know the alginate bead model, it's a system where single cell bacteria are inoculated uniformly into an alginate matrix, where we can make these spherical drops that hardens in calcium chloride. And then the single bacteria form microcolonies, and you often observe a strong size gradient from the outer alginate bead toward the center, due to resource gradients of both oxygen and nutrients. And we've shown that the characteristic aggregate sizes, also the tolerance towards antibiotics, resemble the biofilms observed in infections when you grow it in this alginate bead model. And this is probably also due to the very, very steep oxygen gradients that form in the alginate beads. And if you want to know more details about this model, I will refer you to the paper that was published in 2017 from the group here. Okay, so we grew it in these models, and we recorded thermograms for 3 different growth forms and that were pre-incubated. And generally all the models displayed 2 different peaks, but they varied both spatially and in magnitude between the different conditions. But from these we were a little bit unsure of how to exactly interpret the data. So we tried by looking at, okay, what's the highest peak? How high is the second peak? How long does metabolism go on for? How much energy do they actually use? And none of these sort of predefined quantities could explain differences in the shape or really show the difference between the growth models. So, and we also asked, okay, so what actually causes the stop in metabolism? We show here the accumulated heat. So the area under the curve is the same, exactly the same for all the models. So the total energy released was not different, but by adding additional electron acceptors, the total heat increased. So we concluded that the seasonal metabolism is caused by electron acceptor limitation rather than carbon source. But how can we then investigate overall differences in these large data sets? And there's a nice statistical tool called principal component analysis that you often use for large genetic data sets. It is used to reduce the dimensionality of these large data sets and while retaining a lot of information. And it's very good for emphasizing variation and brings out these patterns in the data. So we did that for all the data points, and we immediately see some clustering between the different categories. And generally the age of the model did not seem terribly important when grown like this, except for the freshly inoculated alginate beads that clustered independent of all others. But this condition is also different from all the others since they are embedded as single cells, but are stuck so they can't swim around in the alginate matrix. So due to this clustering, we asked, can we classify these samples unbiased? So the idea would be to get a machine to say that it belongs to a certain category. So we employed a machine learning algorithm to classify these samples into the different growth models. But these algorithms, they work best with large data sets that capture sort of the true variance of the data. So we used a technique of data augmentation to increase the training data. And we did this by choosing a single data point or data series, and then we used warping functions where we define control points, and from those we can draw random numbers and make multiple iterations and then dramatically increase the data size, sort of artificially. And by doing this, we made the classification algorithm and show that we still have a lot of we still have the same clusters where we trained this model using only one real data point. And so the big dots here, the big circles are the empirical data used for training, and the crosses are all the augmented data, while the small dots are the remaining empirical data points. And we see from this that we could get more than 90% classification accuracy using only one data point. So it seems that we can separate the growth form of planktonic and surface biofilms by using this new tool for looking at metabolism, and also using our new analysis approach. So the take-home message is that we propose a new biomarker that can be used to detect biofilms, and the method we use for it is non-invasive, so we can actually look at the samples afterwards. We don't kill them, and it allows the real-time monitoring of samples, so you can actually check on it, and it's probe-free, so you don't have any genetic markers, or you don't use any stains, etc. And we also developed this new quantitative framework for analysis of microcalorimetric data that is, in our opinion, more unbiased than choosing single entities that you're looking at. But it's important to notice that we do not show any mechanisms behind the differences in these metabolic profiles yet, but only that there exist systematic differences. So further thoughts are, can we use these systematic differences and show if it's still valid for slow growing or non-growing or stationary phase bacteria, and thus if this can be used to develop a method for detecting clinical biofilms. And with that, I would like to thank Novo Nordisk Foundation, who funded this, and Simcell for great technical assistance during the startup of the project, and then the other project members, Kasper, Blaine, Julius, and of course Thomas, and then the rest of the bacterial infection biology group here at the Costerton Biofilm Center. Thanks.
Thank you. Thank you very much indeed, Thomas and Mads. We have quite a lot of questions coming now, so I'd like to move swiftly into the question and answers section. But before we do that, there are a few people who said that they could not attend for the entire webinar. So to those people, I would say, please go and check out our website, where we share as much information and insights as we can on the different applications for microcalorimetry in biofilms. So we have a learning center there. So if you go and explore, and you can ask us any questions that you may very well have. We've also been asked about availability of the webinar and the slides, and we just need to edit together the webinar because there was a bit on the front, so we just need to process it a little bit, but then it will be made available only to the attendees of the webinar, so it won't be publicly available on the web, and so we'll send you an email about that. So please keep your questions coming in, and we'll get to them as quickly as we can. So the first question I have for the two of you is, can you speculate at all as to the difference between the metabolic signals you saw between the planktonic stage and the biofilms? Do you think, can you suggest any reasons for that, any mechanisms?
I guess I can start. I mean, first of all, there's a difference in the geometry of the different models. In the planktonic, at least to start with, you have like free floating cells that are suspended with free access to whatever nutrients and oxygen there is. In the surface biofilm, you have a thin layer of cells, and they will experience a more or less one-dimensional gradient, whereas the alginate beads, you have a spherical model where you have both the size gradient of the aggregates, and then of course a gradient in the resources that they experience. So also a difference in I would say the transcriptional profile within an alginate bead. But as I said, I mean, it remains speculative as to whether as to what mechanisms actually differentiate these. But that is of course something that we're thinking about and wants to pursue.
Great. have some questions about microcalorimetry in general as well. It's a relatively new technique for studying biofilms. I mean, how do you feel about it? How does it fit into your lab and the techniques that you're already using?
Maybe I can start answering that. So we purchased the machine around a year ago, really also with the Department of Clinical Microbiology, to look into antibiotic susceptibility and so on. And also we're going to use it to look into how bacteria are what's it called? Affected by, for example, cytokines and so on, to really better understand the dynamics between eukaryotes and bacteria in infections, and also in for example, the healthy skin. But in the beginning, it was a little difficult because we only got these thermograms and so on. But I think with masses and these guys' work, now we can start to get some real numbers and real associations between what is going on. So I think, and we always have had the problem, we couldn't really measure the different metabolic stages. It was always, are they aggregating, are they not aggregating, are they more or less tolerant towards antibiotics and so on. So I think it provides us, at least it looks like it provides us with a fantastic tool, at least here in the beginning for in vitro observations, but maybe also for looking into different infections. So I foresee that we can really use this on the long term to understand more about bacterial behavior in different scenarios and the microenvironment.
And if I might just add a small point to that, like you can of course measure growth rates and so on in vitro, and you can also have like OD measurements, but I feel like this is one of the few tools that can actually measure the activity unrelated to the amount of cells that you have, which is nice.
Great, thank you. We have a question here about the data analysis that you did. So how do you actually go from the thermograms, which is the output from the cal screener, to the PCA plots? Are there any tools or processes, methods that you use for this?
Yes, I won't go very deeply into all the mathematicals because we have some skilled people that did that. But from the PCA plots, we treated each data point in the thermograms as a variable, so you don't lose any information there. And from that, that's just then you collapse a whole data series into its two principal components.
And also later this week or next week, we'll probably post the manuscript on bioRxiv preprint server, so it's accessible. So that's a plan. We're just working on the last few details.
Great. So metabolic tests can be used to evaluate the effect of anti-biofilm treatment strategies. Do you think that this would be an application for microcalorimetry?
Like, for example, from like in the real setting from infections, I guess that's the only problem, and that's something probably just have to solve, is that to get the sample from the patient into the machine fast enough, because it's not a bedside machine. So I guess if you take a sample, a biopsy or something, you obtain the bacteria, they fastly lose the metabolic fingerprint before we can start measuring in the cal screener. So I think that's the hurdle right now. So that's a task for you guys at SimCell to make it faster.
Mm. So you think it could benefit from being like like in a ward or something? You think that would be useful to kind of have it really close to the patient so you can actually do these tests there? I
would assume that you might. I don't think you will have real microcalorimetric machine in the warts. But maybe if we can elucidate the fingerprints, then there could be some measurements which you can pull out to better understand, and that you could measure for in the clinics much faster. So this can probably be used to elucidate or to look into what is actually going on when bacteria are behaving this way in infections, and maybe devise some diagnostics based on that.
Great. So we have a question very, very linked here about you've used this to look at biofilms in vitro. How do you think it could be used in vivo? So I suppose expanding on the clinical point, could this be used for studying animal models? I suppose it's a bit more tricky, but do you see any way that could be done?
But again, you have the problem that whenever you take the bacteria out of its microenvironment, it changes. So I think it's the machine has to be optimized. So we have to know a bit more about it before we can put in vivo samples. At least we have to try. But I mean, sure, it will be possible down the road with some developments and so on.
But it seems that, I mean, expanding a little bit, that you can already put in organoids and together with bacteria and separate out the metabolic signals. So I guess this would probably be the next natural starting point for getting closer to the in vivo situation. But you could also take just clinical isolates and see, I mean, test resistance patterns and so on.
Yeah. And then I saw in the Q&A, there was another question whether we've done some RNA sequencing parallel to this. And that is also something we're doing right now to try to understand the different peaks, and also when from the machine learning and so on, we can understand where are the differences, and then of course do RNA sequencing based on that again to find out what defines the different subpopulations, or what defines the behavior, the specific behavior of the bacteria in the different scenarios.
Fantastic, thanks. So I have a question here about someone who's working on anti-biofilm metal-based materials, and so they find that agents are active on the center of the biofilm rather than the periphery, and so they're wondering if this technique will help understand something like that. Do you think you can see changes in the biofilm structure using microcalorimetry?
You see the entire heat production. So I'm not sure if you can distinguish between subpopulations because you measure everything. But what do you think, Mads?
Yeah, exactly. So the only way that I would see that work was to simulate the microenvironmental conditions, but in separate wells. So maybe with restriction to oxygen or whatever the microenvironment might be. But it's, I mean, that would still be an approximation. I think there would be other tools for that.
Okay, great. So the data analysis that you showed kind of really gave a very sensitive picture of differences between the metabolism in the different subpopulations there. I had a question here as whether you could use these fingerprints maybe to differentiate, say, a mixed species biofilm. If you had two different species, if you could actually pick out the fingerprints of each one in that mixture, and then could that even be expanded to larger communities with mixed populations?
Well, we know that from the literature that is already published, that different bacteria display differences in their thermograms. So they might have more peaks, or they are not peaking as high, or so on. So the different species definitely have different metabolic features. Whether they can be separated out in poly or multi-species biofilms, I honestly don't know. I mean, yeah, that remains to be tested.
Mm-hmm. Cool. I have a question now about the machine learning model. So the question is, how did you choose the control points that you used to expand the classification accuracy?
To be honest, I don't really know, but it will be in the manuscript.
Great. So we look forward to that.
Yeah.
So do you see how do you see this approach being complemented with metabolomics as a time course?
I think that would suit, I mean, they combine really well, I think. I mean, as said, the thermograms is a bulk measure of whatever is going on a population level in that well, but it gives some real-time information about when you have shifts maybe in metabolism. So it's of course very interesting to see what genes are regulated whenever you have a peak or whenever you have a drop in or shape in the slope of the thermogram. That is something we're definitely very interested in.
Great. So I have a question about the thermograms that you showed as well. When you showed initially the thermograms that looked very similar, there were 2 peaks. Were you able to associate those with any particular feature of the biofilm, a phase of growth or something?
Say that again?
In the thermograms that you showed, there were 2 main peaks that you kind of quite regularly saw in most of the thermograms. Yeah. Were you able to associate the peaks with anything in the bacterial growth curve or any feature of the biofilm, anything that you would have expected to happen? Or was it...
I mean, from the... I think it has already been shown that the initial rise in heat flow is associated with the, I mean, turn on of metabolism, so the exponential phase. And then what causes it to drop, we don't really know, but it's probably going into what you normally call stationary phase. The second peak, we speculate, is probably something to do with running out of oxygen. Maybe turning on or searching for alternative electron acceptors. But again, this remains a little bit speculative.
So we have another question actually related to that topic. Did you try under aerobic or microaerobic conditions to account for the oxygen being used up potentially?
So we inoculated, not inoculated, but loaded these samples the same way. So it's normal oxygenated media without any large pools of electron or alternative electron acceptors. So the only manipulation we did with this was to check what caused the metabolism to stop. And we saw that when we added nitrate, that we had both a little bit of a different shape at the end, but also that the accumulated energy was higher. But we didn't do any controlled conditions with oxygen in these experiments. I don't know if I understood the question correctly.
Yeah, that makes sense to me. So I have a question related to the first part of the talk now. How do you think attachment of planktonic cells is different from attachment of aggregates related to this new biofilm model? Do you think there's any measurable differences there?
Thomas?
Yeah, it's difficult. I don't think so. We cannot really measure single cell interactions. You need, what is it, 10,000 bacteria mass to start to measure.
Yeah, depending of course on the growth rate and then.
Yeah, but potentially if you have a lot of ready to attach bacteria, you can add them, and then. But also another problem is that there is a 30 minutes delay from when you add the bacteria to the wells to load the machine. So the initial attachment happens within that half an hour. So so that we have to think about. I think it would be interesting to study whether single cells and aggregates attached differently? I wouldn't think so, but it's a very, very, very good question. But how to really do it, because you have this loading gap, I'm not sure. Mads?
Yeah, maybe the initial metabolism is different, because you already, I mean, if an aggregate attached, then you already have local gradients in that. Whereas if single cells attach, they need to form biofilm before they actually experience gradients. But yeah, it's a good question.
So, final question: how long do you need to run the microcalorimetry experiments for before you actually are able to differentiate the different conditions?
How long?
Yeah.
Yeah, so we have actually done that analysis, and it seems like at least the first principal component is relatively early. I think within, I can't remember the exact number, but I think it was within 7 hours or something that you have, yeah, the first principal component.
Okay, so that's yeah, and then after that, when you would expect definitely them to be in stationary phase, you see differences in the metabolism. Obviously, you could see that from the thermogram. So, do you see then, if you extend the time, that you can differentiate more, or you can differentiate on different principal component axes?
We've only done two-dimensional principal component analysis. So, I mean, and we've run it over the whole time, which was, I can't remember, close to 24 hours, 30 hours or so. And after that, you don't have much more information, because then there's no more oxygen. But yeah, the second principal component mapped out, of course, later than the first principal. So it seems like that the timing is some of the explanation, but we also, I mean, for the classification algorithm, we use this warping approach that I talked about, where you can sort of make the processes that vary in speed are becoming less important, so it's more about the shape than the actual timing of it.
Great. Well, we're getting very close to time now, so I want to thank you both very much for a fascinating presentation and some really, really interesting answers to some great questions. So I think we wrap up there. I just want to tell everyone watching a few different things. So Thomas and Mads mentioned that they'll be publishing a preprint, so we'll look out for that, and we'll certainly be sharing it on SimCell on the website and on our social media. If you're interested in learning more about microcalorimetry or the instrument that we offer, the Cal Screener, please go to our website. You can have a look around and chat to our applications team and book demos if you're interested in actually trying this out. We know that there's some restrictions in labs at the moment, so we're actually doing remote demos. We can send samples in and we'll run them in our lab. And the final thing is, people have been, as I mentioned before, asking about the recording of this presentation, and we are going to make it available. It's going to be private, so it's not kind of published freely on the internet, but if you attended here, you'll get a private link to the recording of this webinar. So without further ado, I'd like to thank everyone who was here watching, and another thank you to Thomas and Mads, and we'll see you on the next webinar. Thank you very much.